So Lars Muckli is a professor of visual and cognitive neurosciences at the University of Glasgow in Scotland. He's the director of fMRI at the Center for Cognitive Neuroimaging and is also the co-chair of the 7T Imaging Center of Excellence at the Queen Elizabeth University Hospital in Glasgow as well. Professor Muckli earned his PhD at the Max Planck Institute for Brain Research in Frankfurt, and he completed a postdoc at the same institute while helping to found the Frankfurt Brain Imaging Center. His work focuses on brain imaging of cortical feedback, layer-specific fMRI, and multi-level computational neuroscience. Today, he's going to speak to us about multi-level and cross-species investigations of cortical feedback and contextual processing in a talk titled From Man to Non-Human Primates and Mouse, Two Top-Down Streams in Different Layers of Retinotopic Visual Cortex, The Ground Truth. Thank you so much for joining us, Lars.
Well, thanks a lot for the invitation and the nice introduction. Congratulations to doing this the 5th time. I think it's a great achievement. It is also, during these times we have learned to come together in new ways, and I think this is a really fantastic way that works quite well to get this community together. So I'm very excited about this. And let me start by showing you what I will be presenting, a combination of monkey, mouse, and human data, about which I'm quite excited, a multi-method, multi-species approach that we have been able to do within the Human Brain Project. Now I was carried away with the title to say the ground truth, but I was just reading an article from Kendrick Kay who said that layer-specific fMRI has some problems, and it would be best to base models on the ground truth to compare to the data. And I thought that's so true, but that's a great ambition to know what is the ground truth. I remember in the 90s. At that time, with the 1.5T scanner in Frankfurt, I was sitting with Nico Kriegeskorte, and we were thinking about the things one should be doing and shouldn't be doing. And we had some predictions, and we were doing research about apparent motion and the apparent motion streak, the illusion path in between. And I had a clear intuition that there wouldn't be any activity at this apparent motion trace. So we started to do that, and of course, that evolved into several publications around this, that there's a path that is related to the illusion that can be interfered with TMS, that's relevant for signal detection and predictive coding theory. And now in recent years, we showed that that's also relevant for like immediate after a saccade, it moves with the eyes and so on. But the models that we based our intuitions at this early stage was the electrophysiology that was available, and monkey research at that time didn't show any of that kind of activity. So sometimes the ground truth is, of course, an ambition, and it is moving. And this is this example I'm going to show today, I'm especially proud of, because it took me 20 years to find something where fMRI was leading the electrophysiologists to go back and look into the cortex and to actually find something that we have been seeing with fMRI. And now we are in a better place to understand where the signal is coming from. My great thanks goes to Paolo Papale and the team around Peter Roofsema and Matthew Self, from whom the monkey data is, and Koen Segnette and Christian Liefeld, also in Amsterdam, where the data from the mice is coming from. They all used the kind of occlusion paradigm that we have been playing around with for years, and applied those to the animal models. So what we have found, and why do I talk about two pathways? We have found over the years that if we look in non-stimulated areas of V1, we have feedback signals. Some are related to perception, to contextualization of the feed-forward input, and visual illusions. And so this is tightly connected to the perception of input. And then there's another stream which we are starting to see more and more input, which is more related to a more abstract level, cognition, imagery, the internal models induced by willpower to say, I'm going to imagine this, or by listening to something in a different sensory modality and getting activity in V1, or tasks like navigation or mental comparison and simulation related to cognitive questions. And so we'll explore this. In a great opinion paper published in Science last year, Matthew Larkham and his group have summarized some of those intuitions they have about the layers and what they know about the processing in layers of cortex. They have worked for over 20 years with the layer 5 pyramidal cell in rodents and somatosensory cortex. And the idea here is that the feed-forward input that reaches these layer 5 pyramidal cell, it receives its forward input somewhere in layer 4. And in this case, let's say the color of this object. And it's the architecture of these layer 5 pyramidal cells that have two integration zones, one close to the soma and one in the apical layers, which are in the very top layers. So the apical dendrites, thousands of them, are receiving feedback from distant areas and neighboring areas, V2 and so on. And it's those that are able to contextualize the feed-forward input. And so the feedback and the feed-forward input can be integrated there. So that's the idea. So in this case, in this example, the motion feature or the shape feature processed in different pathways would serve as context for the color input into this layer. Now there's two phases of learning. So how do you train this kind of network? How do you train neurons that are disconnected from the outside world? Well, there's this one idea that it takes the first grounding of cortical networks in the to capture the statistical features that are most relevant in the external world. And you can this can take as an example Joseph Feiser's science paper, where they used ferrets and they got trained to external stimuli. So in an early stage, the feature space is more broadly tuned, but later on, if they have no feed-forward input, their feature space is still very tightly connected to the outside world. The analogy Matthew is using is like the ease with which young children can pick up a language and speak perfectly without accent is something that they just absorb the statistics that's outside in the world. The second phase involves a more dynamic learning of context and high-level concepts that establish an internal model to be used in a putative predictive role in the pyramidal cells. So this is more like cognitive tasks, and the metaphor he's using is you can acquire a second language later on by using abstract level of description of the language, the grammar and so on, but you will never learn to fully speak it without accent. And so the second pathway is related, and they have some beautiful evidence for this, to a subcortical network that's feeding in when new associations need to be learned. There's a kind of a memory switch that is turned on to learn associations that then can later be switched off. But this can be learned in an abstract level. So features can be connected that are instructed, and they're not just statistics of the outside world. So we are looking at different examples, and where we looked in visual cortex, and the columns show the amount of visual experience. So first example I'm not going to talk about is blind subjects that we have seen have visual activity in the visual cortex. They have no visual experience, but they have a kind of sense of space that can be picked up. But then there are tasks like amodal completion and illusion perception, which are very tightly related to this first pathway, to contextualize the statistical knowledge about the external world. And then there's other groups of experiments in which we are tapping into visual cognition, imagery, memory, and navigation. So let me start with the classical experiment. V1 gets a lot of feedback from other areas. 10 times more feedback from V2 than actual thalamic input from thalamus. But of course, you know, the thalamic input is the driver, so it's the bully, it's a very loud signal. So in order to tap into the feedback signal, what we have used is we just used very boring feed-forward input, so in this way tuned into the cortical feedback that comes back, and was very pleased to hear that in resting state there's lots of layer 1 signal listening to what other areas are saying. That's exactly the kind of paradigm that we were interested into, listening what the signals that an area is receiving in the superficial layers might be about. So this Markov model going meta-analysis about monkey research shows that there is the feed-forward streams that's more in the around the layer 4 and mid layers, and feedback streams shown in blue that target layer 1, but also layer 2, 3, and the deep layers. So this is the idea here from this model, is neighboring areas would connect more to superficial layers, whereas very distant areas would maybe connect more to the deep layers. What we do here, just for those that don't know the paradigm, and I know I've seen many familiar faces, the occlusion paradigm is based on the idea that we have natural stimuli at which we take one big patch that is occluded, in this case, this white space. We do retinotopic mapping. And then here we have area V1, V2, and V3. We do retinotopic mapping, map out the entire area of the white spot, but then separate the boundary area shown in blue and the center part in orange, to then, in our main experiment, sample the fMRI signal on the different depth layers that we sample equally distance from 6 different layers. They are picked randomly, and they don't refer to actual anatomical layers, even though we use the same number, but that has different reasons. So using this approach, we have one regime in which there's feed-forward input, and then there's one regime in which there's only feedback input, or contextual input, if you like. If we look at the fMRI activity, so the normal way of looking at brain activity, what we see is that in the feed-forward regime, we have more amplitude of activity in the superficial layers, shown here in the rainbow colors. So in the outer layers, we have more activity. In the feedback condition, in these conditions where our receptive fields are on the white spot, we are not expecting any activity. We see almost no increase of activity. Since for this community, I'm going to dive a little bit further into this in a second. But when we look at multivoxel pattern classifier, what we are doing is we take the different streams, different depth layers, and we ask the question, can you use the data from 3 runs to then do brain reading in the 4th run? And in the 4th run, they get the activity from this region that is always white, and guess whether it's a boat, a car, or a market scene with people was the 3rd scene. And you can see that in the feedback regime, it drops to baseline the predictability, so it can generalize the pattern across those 3 runs, learn 3 runs, and then predict for the 4th, except in the outer layers. Whereas for the feed-forward condition, regardless of the amplitude of the bold signal, it can do this equally well across all different layers. That's a sanity check. The steering wheel looks different than the front of the boat, and so therefore there's enough information for the MVPA to pick this up. So again, MVPA information in the outer layers is high for feedback and low in the deeper layers, whereas for the feed-forward, it's high across all layers. If we look at the bold activity, for individual subjects, we started with low numbers. So I'm glad that we can replicate this now, not only across different studies, but also across different species. We show that in the individual subjects for the occluded paradigm, 3 of them show no increase of bold activity during, if you call it resting state, or so, when they are just seeing this white screen. So Tyler's great work when he was here in the lab. I'm just going to touch a little bit so that she can present her data later on, maybe another dinner. But just to highlight one of the most important features what we did. Now we expanded to 24 different scenes, different categories, houses and beaches and so on. And we tried different models to explain the activity in the occluded region. And so they have some information about category, and they have some information about depth level and so on. But the best model shown here for V1 and for V2 is reaching in V2 the kind of noise level. The best model is based on behavior. What kind of behavior did we get? The behavior of asking 50 subjects to do quick line drawings in the region that's here. So if you imagine you're doing the task, you would quickly sketch an image that fits to this image that you think should be underneath. You have never seen the full scene, but you've acquired enough statistical knowledge about the world, how the world should be, that you can draw. And the results superimposed of all these 50 subjects would be randomly gray if it would be, if everyone draws differently. But what you can see is very distinctly the kind of image that you would draw as well. We only took the first strokes of all of those subjects, but actually that doesn't really matter whether you ask for a sample longer or the first strokes and so on. So this is kind of our predictive model that we have, that the visual cortex is actually processing, because it explains well the kind of activity patterns that we find in V1 showing these stimuli. So if you like, it's kind of picking up the statistical knowledge and feeding it down to early visual cortex. This fits to the idea of a predictive processing pathway in which the brain is trying to constantly predict what's coming up and to tune its model so that it is a useful model about the outside world. This scenario in which something is occluded is happening continuously everywhere. So it's a good training set, if you like, for the brain to stay connected and to have good working internal models. So now let's move to monkeys. So I spoke to Peter Rosemer years ago, and he was intrigued, but at some point, he was also skeptical. So then he had implanted in layer 5, deep layers, matrices of 5x5 Utah array or 8x8 Utah array. So many simultaneous recording for which you could do this analysis of not only looking at the activity, but also the decoding of this activity. What they found is the feed forward input is shown in blue, and the non-occluded scene is shown, but in the occluded scene, the receptive field is well into the occluded area. You see the responses summed up over 211 cells, small amplitude, but very reliable, with a delay of 30 milliseconds to the peak relative of the feed forward. So between 42 millisecond rise time peak of the non-occluded and 74 milliseconds of the occluded, there's only 30 milliseconds. And that is quite quick for feedback. That's our interpretation, is cortical feedback, but it's happening instantaneous and very quickly. And that's usually, you would expect that to be ongoing in all these feed-forward stimulation, as well as simultaneously as the feed-forward input is coming. So feed-forward and feedback are not that tightly separated from one. When we talk about feed-forward, we also talk about feed-forward including feedback, and feedback more pure, if you like. So then we took it was shown the monkeys saw the same 24 different scenes, and so we used the human dissimilarity matrices derived from the bold signal during the period, during the block design moment in which they looked at the occluded scene, and that Tyler created that, and we gave that to Peter's lab. And they correlated that from between time points and again, and you can see the dissimilarity. So each row and each column represents one of these stimuli. If like the dark ones are very highly correlated, and those are related to the forests, for example, because the forests are all looking quite alike. And this dissimilarity matrix at the first milliseconds looks unlike the human. But after 100 milliseconds or after 70 milliseconds, it starts to look much like the human. So the interpretation of that would be, since humans see the line drawings, monkeys start seeing the line drawings in the early visual cortex, the mental line drawings, after 100 milliseconds. So now one question we have been asked a lot was, is this just lateral interaction? And we were always a bit open. I think there's lateral interaction in combination with cortical feedback. And my line of argumentation was that it's very likely that it is that feedback is doing a great contribution, but lateral interaction can be excluded. One of the reasons Tyler showed this decay of classification accuracy if you take voxels going more towards the outer. So you see here, this is the occluded area. If we take the light blue and then going to the darker areas, as we take the voxels from those areas that are further and further away from the boundary, the precision, the accuracy drops. And so that interpretation could be that that's related to lateral interaction, but actually it drops in exactly the same way as if we would randomly select the amount of voxels that we are losing, independent of where they are drawn. So there are more voxels close to the boundary and there are less voxels here. But if we just randomly select them, the same pattern occurs, there's no difference. So therefore, the proximity to the boundary does not matter. Now in the monkeys, it's easier to look at that. First of all, the time of at which responses are seen is not different at the small eccentricity versus the larger eccentricity. It's happening instantaneous at the same kind of delay. And the amount of information shown in this here, again, darker patches would mean more information. It's equally, maybe there's a little bit of higher information here, but there's equally drawn information across the entire space. So we can conclude human and monkeys have contextual feedback to occluded areas in V1. Mental, that can be thought of as like mental line drawings, and it takes 30 milliseconds. But there's things we can say from the fMRI data, and that is how is learning amplifying or disamplifying the neurons. We also only know the compartment of 1,000 neurons, 10,000 of neurons. And so we need to take a more focused view, and we can go to mouse research to do that. So here the idea was, if we go back to the layer 5 pyramidal cells, in the superficial layers, there's this integration, and there's this non-linearity that can be seen in rodents when they are tickled at the whiskers, depending on the, so that's around the threshold. So in black, you see the threshold. And Matthew Larkham's lab shown that if you hyperactivate these apical dendrites, they're more likely to detect the tickle. And if they are inactivated, it's less likely to activate. So there's a causal link about the apical dendrites to make them more sensitive or less sensitive to sensory input. And in simulation and more analytical approach, we can see that such a two-zone integration neuron is capable of processing synergies. So like solving XOR problem or making more information than is a simple combination of two inputs. So that's very important if you want to be able to do synergetic or creative brain processes. So let's look at the mice data real quickly. So whole field images, you see the staining across the different layers, but the data I'm going to show you will be from layer 2, 3. So the top layers of cortex. You can see here the different neurons responding now to visual stimuli, and each of these are now a region of interest looked at, and we will see, we look at their receptive field. Again, the same stimuli will be shown as in the previous experiments. Receptive field will be measured. Here is the receptive field mapping. As in both other studies, human and monkeys, we exclude all the receptive fields that are too close to the boundary of the occlusion, so 1 or 2 degrees, and only the inner part shown in red. And so those receptive fields we are looking now in these two regimes, when the full stimulus is presented or only the occluded. So in these snake images, you can see the onset of the full image, and you see a clear response of all those dots, but you also see a response to the occluded situation. Now we show them in a histogram, and you see the response for the full stimuli in blue and for the occluded in red. Again, this is shown the spiking activity. And again, you can see a 30 milliseconds delay between the first peaks at the second wave that comes at non-feed-forward stimulated regions that now receive occluded contextual information from higher areas. What we have been able to do here, what they have been able to do here, is to test those stimuli that the mice had prior exposure to and had learned the kind of image, and those that it hadn't learned. So what does learning do to the stimuli? This is in the left side, we see pre-learning, so stimuli that have been used during training a lot, and post is introduced during the experiment, but then when they acquired, when they have been exposed to more learning. So you can see that the stimuli, so they haven't been exposed, then they are new ones. They're responding very strongly to the full stimuli, but less so to the occlusion. And over time, so over the training regime, post-training, they are now showing a very strong response to the occluded areas, so they can fill in the information and have a reduced or more sparse coding for the full image that they are now very familiar with. So the last slide on this study is we looked at the different neurons, whether they are, how they are tuned. So if a neuron has learned to respond to the full image and the occluded image, it would be along the diagonal. But what we see along the diagonal, so maybe it picks up over learning to be along this diagonal. But what we actually see is two populations. So one that is responding vigorously before the training to the full stimulus, response to the full along this axis. And then another population that after the training is responding, but only to the occluded stimuli, not to the full. So those are two separate populations that we need to think of. And that is new information that we didn't know before. So adjacent to one another, there are pyramidal cells, ones that are representing more and more sparsely after training information that is outside, that is now familiar, and new population of neurons that are now responding more vigorously to the information that isn't shown, that have learned something about this. So one way of thinking about this, there's a model in the about figure ground segmentation, in which here are pyramidal cells and they have inhibitory interneurons surrounding them, and for the figure ground you have an enhancement of the response in the pyramidal cell coming by a disinhibition of the SOM regions. So whereas in the ground where you have a small response, the pyramidal cell gets tuned down because you have feedback to the apical dendrites here, but you also have feedback to the VIP cells that then inhibit the listening to this feedback. So it's kind of an inhibition that is happening here in the background, but in the foreground, you have an inhibition of this channel, which creates a disinhibition and a tuning to this feedback signal. So in our case, we think something like this must happen as well. So before training, you have a drive from the pyramidal cell drive from feed-forward information, but for some neurons, they pick up learning by being now apical driven. So they disinhibit the pyramidal feed-forward input, and they are listening or tuned more to the cortical feedback. So the second pathway, just real quickly, goes to imagery and illusion. And we look at a region that is not stimulated. So this patch in cortex is responding to an illusion, the neon color spreading in this case. And we have a illusion case and then a combination of illusions and amodal completion. So now we have something that is occluded. How does it relate? And a control stimuli. So again, we find in the outer layers, those two conditions can be decoded. And what we are decoding is the color. Is there information about green or red? Is there information about green or red? and that information can be decoded only in the outer layers. So that makes sense to the contextual. But now in the same experiment, so it's a replication of our previous data and a new data set. But now in the same patch, we asked the subjects to imagine, to use the memory, so to say, to do imagery versus perception. We show them in some trials green or red patches, and then ask them in other trials to imagine them. This is all intermingled in the same run, same experiment, in the same boy. And we can see in pink that now deep layers are showing decodability in the deep layers for the content of what is imagined. Is something red or green imagined? The baseline here, the comparison is perception. Of course, that's close to ceiling. That always works across all layers. But the imagery is now more in the deep layers. The last experiment I'm going to show is a collaboration with Javier in Frankfurt and Yeli's lab. Here we asked the question whether memory can contribute to the richness of the occluded scene. So if you have a scene like this and you haven't ever seen it before, you still have a prediction what should be here. This is the concurrent driven information. You have to process somehow the occluder, and at the same spot you also could be expected to process a memory trace in case you have seen the full scene before and have a more richer recollection of maybe some of those features that are hidden from the image and you can infer from the first sight seeing. So our design here was to ask whether episodic memory contributes to what can be filled in. This paper is in print now in Neuroimage. You can look at the details. It's also a bioRxiv paper online available. Just to cut a long story short, on day one, we combined, let's say, a bathtub with a condition, a kitchen for some subjects. And then on day two, we presented a occluded version of this scene and asked whether we can decode now the content, so the bathtub versus other content. And what we found was that we were able to pick up in V1, V2, and LOC the content of the occluded image that they had learned on the day before. So this gives rise to the idea that there's, you can highly abstract, combine things or use episodic memory and see this filled in information. Another trials, we had stimuli that they hadn't seen before, like the bathroom, but have asked the question whether semantically related items like the bathtub are processed or can be read out of the occluded area. And that does not work in V1 or V2, but it does work in LOC. So that's the difference, our difference in the episodic and semantic memory. Episodic memory can be picked up even all the way down to V1. We have another example of navigation where we see V2 and V3 being able to anticipate the next room people are going to if they're familiar, and preliminary layer analysis, but I'm a bit over time, so let me finish by just summarizing the first pathway. Contextualization of input happens in the superficial layers in mice and humans, and in superficial layers, and we have evidence in layer 5 pyramidal cells and monkey. There will be more data coming up about layer 5 cells in mice, which aren't fully matched and analyzed yet. But the idea would be that it is received in the superficial layers and trickles down in the layer 5, but might be outnumbered, might be stronger in superficial than in level 5, layer 5. So mice, monkey, and humans contextualized, learn stimuli in occluded areas. Feedback happens relatively quickly in 30 milliseconds, can be thought of as like mental line drawing. It's not explained by lateral interaction. It's likely to be related to the figure ground amplification and disamplification mechanisms that have been explored by Lisa Kirchenberger in Peter Rosumer's lab. It's learning dependent, and it changes the population of pyramidal cells that are responding. The high-level conceptual is difficult to do animal experiments yet, so this is more human data. We can show that image content can imagine content like red or green content, and we can read out that in deep layers. And in occluded regions of V1, episodic content is represented. And maybe now that I'm here, I remember I should have introduced this. Of course, this memory switch happens here, but there's this collateral that feeds into these deep layers, and that's the effect that we believe that we are seeing in this imagery, that hippocampus and amygdala are driving this kind of memory into these areas. And with this, I thank you, but I thank first my collaborators. And this whole approach is heavily depending on all these collaborators. So give me just a few moments to thank them. So first, there's the, around Tyler and the occlusion paradigm. There's Lucy, there's Tyler, there's Luca, there's Fraser, and lots of my lab are working on related paradigms using slightly variations of that I haven't shown today. Then there's the mouse lab, around the monkey lab around Peter Rofsemer and Paolo Papale, and Matthew Self that provided the monkey data. There's the mouse data from Christian Liefer's lab and Karen Segmente. Of course, the early data is collaboration with Minnesota, Camille, and Maastricht. Rainer and Federico, especially Federico, have been driving this initial start. I'm grateful for them, and thank you for your attention.